Hey everyone, and welcome to The Explainer. I am so thrilled you're tuning in. Today, we're jumping right into this fascinating journey of refinement through the male gamete pathway inside the IVF laboratory. When most people imagine in vitro fertilization, their minds immediately jump to the egg, or maybe the embryo. But before fertilization can ever even happen, the sperm sample has to go through its very own, highly controlled, complex laboratory journey. It is far, far more than just a basic wash.
Okay, let's dive into this. Here is our roadmap for today. We're going to cover the lab as a biological filter, intake, identity, and liquefaction, gradient and swim-up techniques, prep goals for IVF versus ICSI, and finally, severe male factor cases.
Section 1. The lab as biological filter.
We really need to bust a massive myth right out of the gate here. There's this idea out there that a collected semen sample is just, tossed into a dish with some eggs and nature just takes its course. I want to ask you directly, is raw semen actually ready for IVF? Think about it for a second. The reality might completely challenge your assumptions about how these very first steps of IVF begin over on the andrology side. Because if egg assessment is all about asking, okay, which eggs are ready, sperm processing asks a totally different question. From this entire sample, which specific sperm are most suitable for fertilization?
The answer to that earlier question? Emphatically, no. Raw semen is literally never used directly in standard IVF lab practice. A raw sample is basically swimming in seminal plasma, debris, and non-sperm cells. It might actually surprise you just how little of a raw sample is highly functional sperm. Even within the sperm population itself, you've got dead, immotile, or poorly motile sperm, and sometimes leukocytes or other contaminants. So the laboratory acts as a super strict biological filter. Its entire purpose here is to strip away those unwanted components and isolate only the absolute most usable, high-quality fraction. It's a very active process of refinement.
Section 2. Intake, identity, and liquefaction.
But before any of that biological filtering can actually happen, we have to start with a highly controlled, strictly protocol-driven intake process. The whole workflow kicks off with these critical early steps. Number 1 is receipt and witnessing. In the IVF lab, identification is absolutely paramount. Sample identity has to remain firmly locked to all tubes and dishes from the very first second it arrives. Step 2 is liquefaction, which we'll look at in just a second. And step 3 is the initial basic assessment. This is where the lab checks volume, concentration, and motility just to figure out exactly how strong the sample is, and what preparation method is ultimately going to be needed.
Now, what's really interesting about this early stage is this hurdle called liquefaction. Fresh semen isn't always immediately this uniform fluid. It starts out viscous. It physically has to undergo liquefaction to become a liquid that the lab can actually handle, mix gently, and assess under a microscope. If a sample fails to properly liquefy and just stays super viscous, well, that can immediately complicate the lab's strategy for sperm recovery and interpretation. So even before the formal preparation starts, the raw physical behavior of the sample is already dictating the lab's decisions.
Section 3. Gradient and swim-up techniques.
So, how do we actually separate the best from the rest? Well, the lab mainly relies on two primary physical methods. First up is density gradient centrifugation. Think of this as a forced cleaning process. The sample gets layered over this dense media and spun down in a centrifuge. The highly motile, structurally better sperm physically push through the layers to the bottom, while the debris, dead sperm, and round cells get trapped up above. Alternatively, we have the swim-up technique. This one is less about forced layering and more about just letting the best swimmers declare themselves. Motile sperm actively migrate upward out of the semen and into a clean medium that's been gently placed on top. Gradients are fantastic for cleaning up a messy, suboptimal sample, while swim-ups are perfect when you're starting with a pretty good sample and you just want to pull out a highly motile elite population.
Section 4. Prep goals for IVF versus ICS.
Okay, right about here in the andrology workflow, the preparation path drastically diverges. It all depends on the fertilization strategy the clinic has decided on, because the endpoints for these two procedures are totally different. Honestly, think of it like an army versus a special ops team. Conventional IVF, that needs the army. The lab has to produce a high-concentration, highly motile sperm suspension. Why? Because the sperms still have to perform their natural functions. They've got to bind, penetrate, and activate fertilization entirely on their own in the dish. ICSI, on the other hand, is your special ops mission. ICSI stands for intracytoplasmic sperm injection. The lab doesn't need millions of sperm for this. It just needs to find individual elite cells to inject directly into a mature M200 site. And it is absolutely critical that it's an M2 oocyte, meaning it's fully mature and ready for fertilization, rather than an immature M1.
I absolutely love this quote from our source material because it just hits the nail on the head regarding this paradigm shift in the lab. For ICSI, the question is not, are there enough sperm to fertilize naturally in a dish, but can the lab find viable sperm to inject one by one? That mindset completely changes the threshold for what makes a sample usable. A sample that is considered, frankly, terrible for conventional IVF might actually be perfectly adequate for ICSI just as long as the embryologist can find a handful of viable sperm.
Section 5. Severe male factor cases.
So the crucial point is, what happens when biology falters? The numbers are incredibly low and the lab has to handle truly difficult samples? Well, the lab has to adapt aggressively. If a patient has oligospermia, which means a very low sperm count, the lab has to dedicate serious time for a highly intensive search, and ICSI definitely becomes the preferred route. If it's asthenozoospermia, which is poor motility, standard prep methods might be modified so they don't accidentally lose any of the very few viable sperm that actually do exist. And for surgical retrievals, we aren't even dealing with an ejaculated sample anymore. We are dealing with actual testicular or epididymal tissue that requires precise mechanical dissection, and we go in expecting very low motility.
Just to put that into perspective, consider the timeline for a surgically retrieved sample. At 0 hours, the surgical retrieval happens. By hour 1, the lab is doing that mechanical tissue dissection. By hour 3, embryologists are locked into a hyper-intensive, sometimes physically grueling viability search under the microscope. It might literally take 4 hours or more just to select that single rare elite cell for ICSI. This proves, without a shadow of a doubt, that andrology is far, far more than a simple wash. It is an exhaustive, deeply technical hunt.
But, we really have to ground ourselves in clinical reality here. Sperm preparation undeniably narrows the field, but it does not remove all uncertainty. The biological filter we've been talking about, it greatly improves the opportunity for success. We can enrich for motile and visually better sperm, but routine lab selection cannot directly assess the underlying genetics of that sperm. A beautifully processed sample can still result in failed fertilization. We can massively improve usability, but we cannot completely erase underlying biological limits. Which leaves us right at the threshold of the next grade phase. We've collected, assessed, liquefied, spun down, swam up, and selected the absolute best candidates. But the ultimate question remains, if preparation successfully selects the elite sperm, what really happens when it's finally introduced to the oocyte? Does the magic happen naturally in the dish, or does it require the absolute precision of a microscopic needle? Well, that is where the gamete preparation story ends and the embryo story begins. Thank you so much for joining me on this explainer. Keep feeding your curiosity, and I will see you next time.